All right, in part 2 of our chapter on viruses, we are going to be looking a little bit more on how viruses reproduce. I can get my pen to work here. So we're going to look at how we can grow them, like in the lab, and identify them. But then we're really going to look at viral multiplication, including the lytic cycle and the lysogenic cycle. And then we'll talk a little bit about how some of the animal viruses, what happens there in terms of reproduction, because that's a little bit different. So again, just to remind you, a bacteriophage or bacteriophage, however you pronounce it, is a virus that infects bacteria. Okay? We can grow those in a liquid suspension of bacteria, and then we can grow the bacterial cultures on a solid media, like on an agar plate. Usually they have these special little they're like flasks that sit on their side like this, and this is where you'd have the agar, and the bacteria would grow on here, and then wherever the viruses are infecting them, they would just like create little spaces there, or little plaques. So if you saw it from a top view with a microscope, here's your little flasks. You might have little bare patches here where the viruses have actually killed off some of the bacteria and made blank spaces. Think in terms of almost like your zone of inhibition with your antimicrobial lab, but it's the viruses that are actually, as they're destroying the bacteria, it actually just destroys that area on the agar and leaves these blank, clear little plaques in the agar. In terms of when we try to grow viruses in the laboratory, the three main ways we can grow them is we can use living animals, such as mice, rabbits, and guinea pigs. Okay, not all viruses can be grown in animals, such as HIV. We can also use embryonated eggs. All right, they just drill a tiny little hole in the shell, and then they add the viral suspension to that. And this used to be the most widely used method in the past, and it's still used today to grow some viruses for making vaccines. And that's why it would take so long to make vaccines in the past, because you'd have to like grow these viruses in the egg, and so it took a while to do that. So now, of course, they have more, as we saw with the coronavirus vaccine, they have methods now of speeding that up a little bit more. We can also use cell cultures, and this is the preferred method, and this is the one that's used most often now. And that just means that the cells are grown in a culture media, and then we can grow the viruses in those cells. And there's three basic ones here. A primary cell line is derived from tissue cells. These tend to die out after a few generations, so these cannot be used for very long because they die out. Diploid cell lines are developed from human embryos. They can be maintained for about 100 generations. And then continuous cell lines are transformed cancer cells. They can divide indefinitely. That means they can go on and on and on. That's why they're called continuous. And one of the most famous ones are the HeLa cells. This came from Henrietta Lacks. If you've ever read her book, it's very good. Sorry, I didn't mean to bump that. All about, she had, I think it was cervical cancer, and she came, poor family, African American. They took her cells really, and didn't even let the family know that they took her cells, and they've been using them all these years for cancer research and other types of things. And the family only recently found out about it, so it's just kind of it's a really good story. It'd be worth your read if you have the opportunity. Viral identification. Again, remember, you need electron microscopes. It's not like you can just, look at a virus and say, oh yeah, this is what it looks like. So you really can't use morphology as a means of identifying them up close because it takes too much to do. So one of the main things we use is serology. Again, that's where we look at reaction to antibodies. So the virus can be detected and identified that way. And that's part of what they were doing with people who maybe were asymptomatic and had COVID-19, but they weren't showing any symptoms. They could test people's blood and find out, oh, you have the antibodies against the coronavirus, so you must have had an infection in the past. So they actually can use serology to identify that. Remember, viruses are so tiny, so just like what we can do with DNA and other cells, if we don't have very much, we can amplify the DNA or RNA in the virus to aid in identification. So we use that polymerase chain reaction, that method of making lots and lots and lots of copies in a short amount of time. But the main part of this last part of that we're going to go through with viruses deals with how do viruses multiply. Okay, we know they need a host cell in order to multiply, but how do they do it? And there's two main methods. And we're going to talk about, this is in bacteriophages. These are in the viruses that infect bacteria. The two main ways that they multiply is through what's called the lytic cycle or the lysogenic cycle, or sometimes a combination of the two. Lytic cycle gets its name from lysis. That's where the cell literally bursts open, the contents spill out, and it causes death of the host cell. All right? With a lysogenic cycle, the host remains alive. The virus is latent. Okay? Or dormant. Well, that's probably another way of saying it. Okay? So it just keeps reproducing in the bacteria, but nothing happens to it until it's ready to kind of bust out. So the lytic cycle, and you'll need to know these stages, starts with attachment. The virus attaches to a receptor site on the bacterium. Remember we talked about with the bacteriophages, those little legs almost that come down. They would have specific receptors on those that would be attracted to the receptor sites on a bacterial cell wall, and it would hook on there. Then we have penetration. The phage is going to release lysozyme, and that's going to break down part of the cell wall. Okay, it's an enzyme that just deteriorates the cell wall. The bacteriophage then can inject its DNA into the bacterium, almost like a needle, like a hypodermic needle. From there, we have biosynthesis. That's where the phage DNA is going to direct the synthesis of the viral nucleic acid and proteins. It doesn't mean it's building a new virus yet. It's just building the parts. Okay. Oh, we're building the protein coat. Oh, we're building the DNA or the RNA. It's in maturation that the actual DNA and capsids are assembled into complete virions. Those infectious particles now that are all like almost a mature virus. Okay. So it's not till maturation we actually get them all assembled, right? And sometimes this stage is called assembly too, just because that's exactly what happens. Finally, we have release. The newly assembled bacteriophages are released due to lysis, or the breaking open of the plasma membrane. And again, lysozyme is there again, which breaks down the cell wall, so we can fully break open that bacterial cell so that those bacteriophages can now infect other bacteria cells nearby, and we start all over again. Okay, so attachment, penetration, biosynthesis, maturation, and release. Oops, sorry about that. I was just trying to slide my slide up here. So here's just kind of a visual picture. There's lots of different diagrams you can look at, but again, it's docking at these receptor sites in attachment. Here it's injecting the DNA inside in penetration. Here it's just making the different parts. They're not assembled yet, but it's making the parts in biosynthesis. In maturation, here now we're assembling them into fully mature virions or mature viruses. And then here it's busting open the plasma membrane and the cell wall in lysis for the release. And that's how it gets the name the lytic cycle. The lysogenic cycle occurs with what we call lysogenic phages or phages. These are temperate phages. They can proceed through the lytic cycle, but they are also capable of incorporating their DNA into the host cell's DNA. And that begins what's known as the lysogenic cycle. So lysogeny, that term by itself, means that the phage remains latent or inactive. Okay, and then the bacterial host cell is called a lysogenic cell when it's in that state. When the viral DNA is part of the bacterial DNA, but it's not doing anything. It's just hanging out there. So we still have attachment. The phage is still going to attach to the bacterial cell wall. It's still going to penetrate and inject its DNA into the bacterial cell. But then, how we start to have a little difference here, the phage DNA is going to form a circle. That circular phage DNA is going to recombine and become part of the bacterial cell chromosome. Once it's in this stage right now, it is called a prophage or prophage. Okay? Most of the prophage genes are repressed by a repressor proteins. They're blocking those operators. And so if you think back, it's almost like when we talked about the inducible operon, it's normally in the off position because repressor proteins are blocking the operator. So our RNA polymerase can't come and transcribe and make more RNA. Well, this is kind of the same thing that's happening here, is that this is what keeps that prophage in a latent or inactive dormant state, is that those repressor proteins are blocking so that there can be no transcription translation of proteins needed to make the capsids or anything else going on in that cell. Okay? Every time the host cell's chromosome is replicated, the prophage DNA gets copied too, right along with it. And that prophage is going to remain latent until some event. We don't know what will trigger it. Sometimes it might be UV light. It might be some other event, but it causes the excision or removal of the prophage, causing it to go into the lytic cycle again. And then it will follow all those other steps of biosynthesis, maturation, release. So you have here, it's kind of a bigger picture here, but it just kind of shows that the phage DNA attaches to the host, injects its DNA, so it attaches and injects. And now we have two different things that can happen. We can have the phage DNA is forming that circle, and it's either can go into the lytic cycle over here, where it's going to go through biosynthesis, maturation, and release, or it can now go into the lysogenic cycle. So over here's the lytic cycle, over here's the lysogenic cycle. And so it incorporates its circular DNA right there, right into the bacterial chromosome. So every time the bacteria divides, so does that gets copied as well. So when the bacteria is going to replicate, that viral DNA gets replicated too. And just going to keep going. It's going to just keep going back and forth, making more and more and more cells that have this viral DNA in it now. Something will trigger, okay? Some event is going to cause it to be excised or cut out. So now here it is being removed. And then what happens, once it's removed, it can go straight down in here and enter the lytic cycle and go through biosynthesis, maturation, and release. So some phages may just totally do the lytic cycle. Some phages, called temperate phages, may do the lysogenic cycle for a while, and then when some event causes them, they'll switch and go into the lytic cycle and destroy the bacteria. But you can see the bacteria isn't destroyed here. It just keeps reproducing until something triggers it. with animal viruses, they also have attachment. They have receptor sites on the animal cells. And sometimes with enveloped viruses, the spikes act like attachment sites to attach the cell plasma membrane. They have entry, but the forms of how they get into, they don't necessarily just inject like a little bacteriophage would do. They enter by endocytosis, specifically pinocytosis, where the plasma membrane pinches in and a vesicle forms around the virion, and it's brought into the animal cell that way. We can also have fusion. That's where the viral envelope fuses with the plasma membrane and releases the capsid into the cytoplasm. And again, that capsid is going to have its DNA in there, inside it, to put that viral DNA inside the cell. But a new step that we have here is uncoating. This is where you have separation of the viral nucleic acid from the protein coat. Okay, so you're basically just removing that capsid and digesting it, and then all you're left with is your viral DNA or RNA that now can get into that animal cell. Then we have biosynthesis with DNA viruses. They will replicate their DNA in the nucleus just like we do in our normal animal cells. The capsid and other proteins are synthesized in the cytoplasm, I'm sure on the ribosomes, and then they migrate to the nucleus. RNA viruses multiply in the cytoplasm. Okay, they multiply in the cytoplasm. For maturation, the viral DNA and the capsid proteins assemble to form those complete viruses. And release, they can either rupture the plasma membrane and release it, pretty much almost like in the lytic cycle. It almost works the same way. Or they can do budding, and that's when the plasma membrane forms an envelope around the capsid, and it just becomes part of it, and it just kind of releases it that way. All right, so that's all we're going to cover for this chapter. We're not going to cover the prions or the viroids. We're just going to cover viruses.